Yeah, good afternoon, everybody. Thanks for joining. I'm Peter Bauer, Chief Medical and Genomic Officer at Centogene, and I'm happy that you made it today to join the scientific webinar on precision medicine and Parkinson's disease. I will just wait another minute or so that we have the majority of the registrants aboard, and then I will start sharing my slides. And so, welcome once again. Let's kick it off. So, I'm excited to have this 45 minutes with you today to share an update of what we at Centogene have been doing in the last couple of months towards the multiomic era in Parkinson's disease, which should enable precision medicine in a neurodegenerative disease that still doesn't have a cure, but just symptomatic treatments. And I've prepared today an introduction into the disease, especially what has changed recently also in our understanding how we should diagnose Parkinson patients. I want to share with you an update on the genetics in Parkinson's and our ROPAD experience, which is a large observational study we have kind of conducted over the last 5 years. And from there, which type of multi-omic analysis we could already perform in this observational study cohort. And also will give you time to ask questions and ideally answer them within our webinar. So I will really keep it short as an intro. I think the clinic is clear. We face a neurodegenerative disease, and we also know that this is quite prevalent, and a percentage of the elderly population will face Parkinson's in their life. And just to give you some numbers that are already 20 years old, but show how frequent we have movement disorders in an aging population, I've brought this Lancet Neurology paper of Gregor Wenning and co-workers with me, where you can see that signs and symptoms of Parkinsonism will be seen in 6 to 8, almost 8% of the elderly population, more in men than in women. And overall, we see really that more than 5% will face movement disorders with Parkinsonian features at later age, higher age. There's also some symptomatic forms of Parkinson's, but the idiopathic Parkinson that we are talking of specifically represents 2/3 of this population. There's other movement disorders, and tremor is even more frequent, but as you can see also the subjective disease severity is very different there. And so it's, I think, the most frequent severely impacting neurodegenerative movement disorder. Now, what really has changed in the same 20 years since this publication was set forth is our understanding how we should define a neurodegenerative disease. And most of the progress was made, I believe, in Alzheimer's disease, where the clinicians and the scientists have started years ago to separate the syndrome, which is the clinically identified impairment, from the biology, saying the driving factors that might even be present in an individual the whole life. And there we've seen, and that's the most recent publication also from Alzheimer's and Dementia from 2024, that the field completely started to define the disease by its biology. This means you need a specific neuropathologic change with biomarkers. This is imaging, but this is also blood-based or CSF-based biomarkers, and with defined thresholds and observations, the disease is diagnosed. And these disease-specific core biomarkers will even be seen in people that are still asymptomatic, which means we start to see the development of disease even before individuals notice themselves. Of course, there's still research, and the mechanisms that are involved in the progression are still under research and not absolutely defined. But what is really new and important is that symptoms are now a result of the disease process and are no longer necessary to diagnose Alzheimer's disease, which means even if you don't have symptoms, but you have the underlying biomarkers detected, you are an Alzheimer's patient and not longer at risk of a disease that you already have, which is subclinical, but clearly measurable. And so the other way around, symptoms that we used to take for a diagnosis of Alzheimer's in the past are not diagnostic of Alzheimer's disease if those biomarkers are not also demonstrated. So there is other conditions that mimic the disease. And so we have, with the same biology in the end, even different phenotypic presentations. But the biological definition of disease was a big step forward, which was also paralleled in similar discussion and developments in Parkinson's. There, we are not so advanced yet, and I will share with you why in this intro, and then look also how multiomics can help us to bridge the gap that we still have. So in 2024, Simoni et al. Also published in Lancet Neurology a biological definition of what they called neuronal alpha-synuclein disease. And in this case, once again, it's a definition of a disease, not of symptoms. It's mainly related to the core biomarker that has been established in the recent years, which is the demonstration of seed amplification pathological alpha-synuclein in cerebrospinal fluid. And then you can distinguish between preclinical and prodromal stages and the manifest stage, depending on the symptoms. But it has important implications in the Parkinson's fields because this seeding assay, which is now the main cornerstone of the definition, is also positive in Lewy body dementia, which of course doesn't have primarily Parkinson's symptoms, but the cognitive decline phenotype. And so it's a broader phenotype and a more narrow pathology in an integrated approach, which was also proposed in the paper. There is also aspects of dopaminergic dysfunction and the overall functional capabilities. But again, like in Alzheimer's, the potential impact was to enable defining a more reliable disease cohort for early preventional trials and have also tools to measure the impact of disease-modifying therapeutics. But as I said, in Parkinson's, there is still a controversy, and there is in the same year, in the same journal, also publication by Günter Höcklinger Group and co-workers that at least opened up the discussion that classification of the biology is very needed, but it's not a redefinition of the entire disease as of yet. So they came up with a multidimensional classification framework that also, of course, relies on the seeding assay, the demonstration of pathological alpha-synuclein aggregates, but then on top, signs of neurodegeneration through imaging, and also added what was missing in the other model, the importance of pathogenic genetic variants that are linked in the end to clinical features. Of course, it acknowledges the central role of alpha-synuclein, but also that other forms of Parkinson's, at least currently, lack demonstration of this marker and therefore would then have trouble to be included into clinical programs and be diagnosed as synucleinopathy, as it's proposed in the definition on the slide before. We would avoid a single biomarker disease label, and this also is important to keep an eye on the, I think, still a more diverse pathophysiologic genetic picture that we see in Parkinson's. Again, of course, also this classification aims to have better stratified cohorts and kind of matched target case control setups. This is ongoing, but I think it's worth having an eye on that going forward, where especially maybe better markers are more inclusive for the phenotypes we want to include, and also to study the pathophysiology from the genes into the disease to understand which trajectories of disease progression exist. On our genetic front, we have made great progress in the last 10 years. And this is still a learning example because, as you can see, the majority of the kind of legacy Parkinson-associated genes have been discovered from 1997, where synuclein was the first to be demonstrated, and Parkin a year later into the rarer recessives, and then the more frequent not fully penetrant LRRK2 as a dominant trait. And then around 2010, we've seen the most important risk factor, GBA1, entering the stage, and a very rare one 2 years later. But then we had quite a significant gap from these discoveries until we came across the notion that in the GBA1 gene, there is also a non-coding variant contributing risk, especially in the African population. I will talk about that in more detail later. But this was an almost 1% risk that was guided by a genome-wide association study, so a new approach to find the missing heritability. And just 2 years ago, we learned about a single variant in RAP32 that is interacting in the LRRK2 pathway, contributing to the risk. That was a variant-based risk analysis that could bring us this gene to the list. So what is new in monogenetic Parkinson's is we still have well-established classical Parkinson genes, but we added RAP32, which is now PARK26 in the nomenclature. We also did not see a lot of change in the early onset Parkinson's, which is Parkin, PINK1, and DJ1, the most prominent. And there's a growing list of atypical Parkinsonism that links kind of features of Parkinson with other neurodegenerative features, like in ATP13A2 or in DNA JC6, where we've seen good evidence for genotype-phenotype correlations, but of course assume that also with these genetic abnormalities, we have also distinct pathophysiological patterns to understand. All overall, we could establish a large cohort, which is called ROPAD, and I will explain that in more detail also in the next part, where we did a genetic screening in Parkinson patients, early diagnosed, and we learned that roughly 15% of the reports we remitted contained a relevant genetic finding associated with a Parkinson feature. The most important is GBA1, more than 10%. Second most important, LARC2 with almost 3%. And then all the others are 1% or below. But we also seen a couple of cases where even two diagnoses came together in one individual. Not a surprise that the two more frequent genetic findings can then also be the most frequent dual diagnosis observation. And we looked also deeper in the most frequent ones, GBA1 and LARC2, and seen that risk factors account for 55% of the genetic burden, while Gaucher disease relevant variants, and I will talk on that also later in Multiomics, is 45%. And in LARC2, we have also a very frequent pathogenic variant, which is the G2019S, 80%, and all the other variants with established pathogenic mechanisms account for 20%. Interestingly, now with the new findings of the African variant, for example, we could add another 64 GBA1 Parkinson patients from the ROPAD cohort, which was 12,500 patients in the end, that carried this variant and could be explained after the initial publication. And we could also find that the RAP32 was present in Parkinson patients. Roughly 0.5% of the individuals carried this variant, which closely interacts with the findings in LARC2 functionally. Now, I mentioned the ROPAD experience already more than once, and just to give you the outline of the study, and then especially what we did to understand multiomic data in Parkinson patients based on the samples we could collect. So we started in 2018 with an international study with more than 120 sites that recruited at the end more than 20,000 Parkinson patients. And you can see there was patients from especially Europe, Israel, Turkey, and then North America and South America, where we had a comprehensive data set, including clinical data, medication, motor symptoms, clinical features. We did the Parkinson panel sequencing, later also the exome and genome sequencing, and we received consented dried blood spot filter card samples to generate a biobank and have sample material available also for multiomics. Multiomics, early stage, was mainly doing genome sequencing and biochemical analysis with the especially the enzymes of lysosomal pathway proteins, especially glucocerebrosidase. We also looked into the glucosylceramides, and we also quantified glucosylsphingosine, which is kind of a degradation product of glucosylceramide, which accumulates in Gaucher disease and is quite interesting also to be researched in GBA1 Parkinson's. On top, we realized that we can do RNA analysis from the dried blood spot filter card, and I will share some data on that as well with you. The ROPAD cohort was a very typical Parkinson cohort, and the first phase, ROPAD 1, was published in 2024 by Anna Westenberger and colleagues, 12,580 patients. And as you can expect, it's a cohort where we have a majority of male individuals because Parkinson's is more frequent in males than in females. And we've seen that they had an age at onset around 59 years on average. The clinical diagnosis was done within 1 year. but the enrollment into the study was only 7 years later because genetic testing kind of was also a new paradigm that was tested and offered there. Overall, many of those that have been included had at least relatives, close relatives also with Parkinson's, which tended for a potential enrichment also of genetic findings. It was mainly a European cohort with smaller fractions of kind of other ethnicities. Looking into the data after we've done the genetic analysis, we could see the fraction of the gene-related findings that I shared already a couple of slides ago. But then we could also look at the age at onset that was reported and the genetic risk variant or diagnosis that was established. And what you can see is that also replicating the literature, if you have an early onset recessive genetic form of Parkinson's, your age at onset is significantly earlier than those that are idiopathic or only risk factors with a dominant inheritance. So you see that this kind of goes almost 25 to 30 years earlier than the idiopathic group. And we see also slight significant earlier onset in synuclein positive Parkinson patients, while kind of the idiopathic and those that are caused by GBA1 or LAG2 are not significantly differences. Or you see, although you see that on average, there's a little bit an earlier manifestation in the both genetically driven diseases. Now, we tried to establish more testing in the filter cards we receive from the Parkinson patients than just the genotyping. And for that, this multiomic testing could rely on our capabilities that we can extract also from the filter cards material to quantify enzyme activity or biomarkers. And so this kind of medical device that we provided in the study, but also use in our standard diagnostics, just is an ideal biomaterial archive to do additional biochemical testing. You need a couple of punches there, and then you can test these defined volumes of the blood that has been dropped on the card for biochemical tests. And that's just what we learned over the last 6 years using these samples and applying our tools, especially about differences in Parkinson's disease. Now look, the first we've asked is whether the biochemical parameters we have established for Gaucher disease also work in Parkinson samples and whether we see any differences. So we did first a measurement of the glucocerebrosidase activity. And just to give you an understanding what you can expect, I have summarized the data we've seen in Gaucher disease patients where this test was developed 15 years ago, and non-Gaucher disease patients. And in this cohort, we defined a normal activity with roughly 5 micromole per liter per hour from filter card, which is shown here in thousands of non-Gaucher individuals. And we've seen that in Gaucher disease patients, usually enzyme activity is below 2 micromole per liter per hour. But what you can also recognize is although this is very clear and highly significant, there's huge variation. So we have a variance in these measurements of almost 40%, even in the non-Gaucher individuals, indicated by the arrow bar. And in Gaucher patients, it's even higher. You might ask why this is high in Gaucher patients. And the reason there is a little bit artificial because sometimes you receive a sample of a Gaucher patient that is under enzyme replacement treatment. And then, of course, you have very good performance of this test with the artificial enzyme that he received just a couple of days before the test was done. If you can exclude those, which sometimes is not easy for us as a reference laboratory, you will have smaller variants, but still you can expect that it will not be better than in the untreated healthy controls. Now, if we check the same enzyme activity in Parkinson patients, we did kind of a large study with 3,200 Parkinson patients from ROPAD. And we matched these samples with same sex, same age controls that didn't have Parkinson's. And to enrich for GBA1 genotypes, we looked especially for a cohort, subcohort of 800 of the 3,200 that had Parkinson's with a GBA1 variant. And we had a similar cohort with 812 individuals that did not have Parkinson's with a GBA1 variant. All these GBA1s heterozygous, like the established risk factor. What you can see in the enzyme activities that we could measure is that the GCase activity is roughly 20% lower if you have a GBA1 heterozygous variant. You can see it here from those that have Parkinson's without and with, 20% down, but also in controls that don't have Parkinson's and controls that have only a GBA1 but not Parkinson, 20% down reduction. The overall levels in both arms is identical for those that have GBA1 variants or don't have variants, which tells me that the change looks like be caused by the genotype, but is not influenced by the fact that those in red have been the Parkinson patients. That's interesting because GCase activity is also seen as a driver of Parkinson's, but you see that matched healthy controls can have similar amounts, still not answering all the questions, but at least it's not a diagnostic marker. That's, I think, conclusion from this observation. We looked also into the biomarker glucosylsphingosine, lyso-Gb1, which is a very good marker in Gaucher disease. And once again, just to show you how extremely good this marker works in Gaucher disease, usually here you have also levels in normal individuals around or below 5 nanograms per milliliter blood. And in patients, you have huge accumulations, 100 times more even is possible or below. So this marker shows the accumulation of the substrates that then also correlate with the disease progression here in a newborn with Gaucher disease that build up more and more of this lyso-Gb1. And it could also be shown that under enzyme replacement therapy, levels can go gradually down, showing efficacy of the enzyme replacement. So this marker reflects disease severity and treatment effects. And you can use it for longitudinal studies, which we have shown in a couple of publications with collaborations as well. Now, in Parkinson, the picture is much more minute. And again, you have the same setup. We have here in red those with Parkinson's, either with GBA1 variants or without. And then with without Parkinson's, variant positive, variant negative. And what you can see is if you have a variant, you have 15% increase in lyso-Gb1, also independent whether you have Parkinson's or not. Once again, very similar to the glucocerebrosidase activity picture. You have a reduction in the enzyme activity and an increase in the biomarker, but both are just within 20%, and it's not a massive change. There's no accumulation of this biomarker in Parkinson's disease. I think that's interesting. So not helpful for establishing a diagnostic, maybe still interesting to monitor patients with these genotypes. And we also checked the glucosylceramides, which have been published to be influenced by Parkinson's. And there we've seen, like also in the literature, that the mean value of the total glucosylceramides is elevated in Parkinson's. And here you see that you have roughly 20% more glucosylceramides if you have Parkinson's compared to healthy controls, matched healthy controls, which would establish and thus confirm that glucosylceramides are associated with the phenotype and not so much with the GBA1 genotype, because we don't see the differences whether they have GBA1 heterozygosity or not. So this was kind of a very basic but informative observation in the ROPAD cohort from filter cards doing biochemical analysis. And I want to share you in the next couple of slides also what we learned on transcriptomics. I told you that we are also able to extract RNA and do specific qPCR, or we could do RNA sequencing from the filter card material. In this case, we have been contacted by GP2 working group working on the African variant, because this variant that was found by GWAS analysis, as I told you, is a non-coding variant. So it changes the nucleotide in intron 8 of GBA1. And the question is, what happens after you have here on DNA level a variant? Does it change the RNA? So is splicing affected? And how can this be kind of demonstrated with experimental data? And so one of the speculation was that through this variant part, or the whole intron 8 is retained in the transcript and then producing a protein that is no longer functioning and thereby impairing glucocerebrosidase enzyme activity and also leading to a Parkinson risk. Interestingly, not enough severity to cause Gaucher disease, because we have not seen this variant, excuse me, really contributing to the more severe biallelic Gaucher phenotype. So what we did, we designed a couple of primers that could test the RNA molecules present in patients and controls outside the critical region, but also inside the critical region, and try to demonstrate direct proof of part of intron 8 or full intron 8 retention. And with that, we have produced data in 40 heterozygotes with this African variant. So this is all kind of samples that we could collect in our biodata bank. And we could also collect 74 controls that had the wild type. And what you can see in kind of the quantification of the results is that we don't see a change of the amount of GBA1 RNA, mRNA that is available for the overall transcript GBA1. So the primers that were outside the critical region showed identical amounts after standardization. But when we had specific primers against faulty splicing with intron 8 retention, we've seen that only those, or especially those individuals with the heterozygous genotype, produced an amount of RNA that was not present in controls. It's not that the control are perfectly splicing all the times, but for the heterozygous, we've seen that there's a clear shift, and the abundance of this small amount of misspliced GBA1 transcript was only observed in individuals with heterozygous African variant alleles. So the presence does not alter the overall abundance and turns a small proportion of the canonical transcript into intron 8 containing transcripts. We could then sequence these to these intron 8 retained transcripts and realize that a small fraction is full intron retention, And the larger fragment of this faulty RNA is only partially including intron 8. So this is an aberrant isoform that is caused by the variant or triggered by this variant allele. And then when looking into the splicing signals, we realize that there is a branch point in intron 8 that helps to form the splicing so-called lariats. And this branch point is exactly hit by the African variant, and thereby kind of the proper splicing machinery has some hiccups, I would call it, and produces then a small fraction of either fragmented or full intron 8 RNA transcripts, while interestingly, the majority still works as it should, and 95 or even more % of the transcript looks still very functional. This was interesting, but immediately also triggered the next question. But if we don't see a lot on the mRNA level, do we see something in biochemistry? Is the enzyme still a little bit less active? And do we see also reflection in the lipid profile? And I only share with you kind of the comparison of the glucocerebrosidase enzyme activity experiments. So what we knew already, and I demonstrated you a couple of slides before, is that if we have a GBA1 variant, whether it's a PD patient or a non-PG patients, we have a reduction of GCase of 20%. Now for the African variant alleles, we have added the two samples with either heterozygous or homozygous variants, and you can see that in those cases, also with quite a large variance, which I also explained you technically already in the intro slide for glucocerebrosidase enzyme activity, we see that these reductions resemble those rather mild risk factors that do not cause Gaucher disease and do not have the extent of classical mild or severe Gaucher mutations, which also fits well with the observation that these individuals with homozygous African variants, they have a risk to develop GBA1 Parkinson's, but they don't have dysfunction and peripheral disease like a mild Gaucher disease phenotype. So all in all, I think the multiomic integration of our findings with the genotyping, the RNA work to demonstrate the partial and sometimes full intron 8 retention, and then also the enzymatic activity measurements in the same samples is a great tool to bring together these data points for a hypothesis and to ascertain the importance of a variant and kind of also the impact and compare variants in different of these quantitative systems. So we've done quite some work also on the publication side in the last years with this ROPAD cohort especially, and on Parkinson's beyond. One that I mentioned is contributing to the elucidation of the mechanism of action of this intronic branch point variant in GBA1. We also have published some data on the glucosylceramides that I showed you also, that they are changed in Parkinson's disease, but do not seem to be involved in the disease pathogenesis. At least we don't have positive proof of that yet. We had the large paper on the ROPAD findings in 2024, where all the genetic observations have been shared with the public. And we have done more recently for also the RAP32 linked Parkinson's, where also the patient, the deep phenotyping of the patients and running the seeding assays help to also add RAP32 to kind of the disease genes. Now, to conclude, I think I hope I could share with you that in this prevalent neurodegenerative disease, genetic testing is very important because 15% have a major genetic risk factor. However, the new disease definitions and the need and still the gaps there rely on the availability of robust diagnostic biomarkers. And at the moment, everything is CSF analysis of the seeding assay, which does not yet include all the variants of Parkinson. So I think we might need more markers to come. The dysfunction in the endolysosomal system happens very early, also proven by the genetic forms of Parkinson's that have genetic functional variants in these systems, and is also to be explored for diagnostic and longitudinal biomarkers. That's an initiative that is also very high on the priority list, also for the big research networks. And going forward, the integration of genetics and biomarkers will be used to stratify patient cohorts in translational research, like we've seen in Alzheimer's. It's already happening. And I'm glad that we can share also our part in that story with our capabilities in multiomic diagnostics using the Centocard technology, which opens into genomics, transcriptomics, proteomics, and metabolomics. So at the end, just to quickly share that this is, of course, a big and efficient, successful teamwork. I have to thank many colleagues internally, just to highlight Christian Beetz and Mandy that did a lot on the analysis of the ROPAD samples and the RNA part, especially in the genetic understanding of the variants and the genotypes. We have Tobias and Emma working now also on broadening that. And we have a team in biochemistry, Steffen and Barbara, on the assays. Very efficiently, Krishna helping with the bioinformatics that wouldn't go without. And then the team that managed the ROPAD study, Jeffrey, especially in York, to be mentioned, and also the medical support with George. I work since years with Christine Klein, who is a great also colleague to discuss and move where we have still limitations. And then, of course, I should not forget that all the PIs that participated in ROPAD have been also part of the success story with their patients that have agreed to also do more than just genetics in Parkinson's. The project on the African variant was funded by ASAP through Michael J. Fox, which I also want to mention. I thank you for having joined and wish you a good day. Thank you. Goodbye.